Hi everyone, welcome to Biology Pro. Today a lecture is about proteins. Okay, so what are proteins? Proteins are polypeptides of amino acids, and these are the heteropolymers of amino acids. And your protoplasm, the most abundant nutrient in the protoplasm is proteins. So protoplasm contains 15% of proteins. Next to the water, highest nutrient in the protoplasm is your proteins. Okay, and proteins are composed of elements like carbon, hydrogen, oxygen and nitrogen. Most of the proteins, sulfur is also present in most of the proteins, right? Next, protein structure can be studied under 4 levels. Those includes, number 1, the primary structure, secondary structure, tertiary structure, quaternary structure. Right? So structure of the proteins can be studied under these 4 levels: primary, secondary, tertiary and quaternary. So let us find out the first one, primary structure. What is it? Primary structure explains the sequence of amino acids that are arranged in a polypeptide chain of proteins. That means what is the first amino acid, second amino acid and so on. This sequence is explained in the primary structure. This is an amino acid, serine, this is cysteine, tyrosine, glutamic acid. Imagine this is one sequence in any polypeptide chain of a protein. So how these are all arranged, this information is present in the given by the primary structure. That means the positional information, these amino acids must be present in this way. So always the primary structure of a protein is a linear chain of amino acids. These are all the amino acids, and all these amino acids in a protein molecule are connected by the peptide bond. So this is the first amino acid, which is called N-terminal amino acid, and the last amino acid is called the C-terminal amino acid. Because your left side is called N-terminal and right side is called C-terminal. So left side N-terminal is called the first amino acid and C-terminal is with the last amino acid. That's what the primary structure of a protein molecule explains us. So to understand better, imagine your primary structure is like this, which is a rigid rod. This explains, all these boxes will show you what is the sequence of this amino acid. That is what the primary structure explains. Okay, right. So next, every time the protein does not exist as a rigid rod like this, some portions of this rod gets folded, and so this is called the second level in the structure of the protein. That is called the secondary structure. How this happens? In secondary structure, the protein thread is folded in some portions. Due to this folding, right, the protein can exist in either a alpha helical form or a beta plated structure. So how protein molecule gets this alpha helix and beta plated structure? This is because of hydrogen bonding, and this hydrogen bonding could be intramolecular hydrogen bonding, or it can be intermolecular hydrogen bonding. Intermolecular hydrogen bonding. So we'll find out what exactly these two are. So secondary structure of a protein molecule can have 2 types of bonds. One is peptide bond, which is the strongest bond. In addition to this, there is a hydrogen bonding can also be seen, right? So first is your alpha helix. What is it? Alpha helix of a protein explains that it is a spiral which is formed due to intramolecular hydrogen bonding. What is it? Imagine this is the helical structure. Imagine this is your protein thread, right? And sometimes the protein thread is folded at different places in this way. So you get an alpha structure, you get an alpha helix. Your alpha helix is like this because of folding. So this folding is due to the intramolecular hydrogen bonding, because this is a single polypeptide chain. In this single polypeptide chain, what all the amino acids are present, so the intramolecular hydrogen bonding exists among those amino acids. So as a result, the protein gets a shape of helix, right? So this is your helical form. How this happens? So imagine, just look at into this picture. This is one amino acid. This is the carbonyl group of this amino acid, and this is the amine group of the other amino acid. So there is a bonding between the carbonyl group of one amino acid and amine group of other amino acid. This is called the intramolecular hydrogen bonding. You can see in all these positions there is an intramolecular hydrogen bonding. Due to this intramolecular hydrogen bonding, the protein polypeptide chain becomes helical, which is called alpha helix, right? So this bonding is present between the first to the fourth amino acid in a polypeptide chain, and this is one turn is with 3.6 amino acids. Okay, so this is what is alpha helix, and this alpha helix, almost all helices are right-handed helices, and alpha helix structure of a protein is found in keratin. So because of this alpha helical structure, the intramolecular hydrogen bonding, keratin is the strongest protein. Second is beta plated structure. How this happens? In beta plated structure is due to the intermolecular hydrogen bonding between two segments in a polypeptide chain or between the two polypeptides. These are the two polypeptides for our convenience, and this is the hydrogen bonding between the carbonyl group in one amino acid and the amine group of the other amino acids in different polypeptides. This can be intermolecular hydrogen bonding, and due to this intermolecular hydrogen bonding, a protein can have a beta plated structure like this. This is due to the hydrogen bonding between two segments of a single polypeptide, or if a protein is with two different polypeptides, you can find out this between the two different amino acids of two different polypeptides, which may be a intermolecular hydrogen bonding, right? Now this intermolecular hydrogen bonding gives a protein a beta plated structure. Fine. So beta plated structure can be seen in fibrion protein, right? So like this, the secondary structure is because of hydrogen bonding, which is intramolecular hydrogen bonding or intermolecular hydrogen bonding. If it is intramolecular hydrogen bonding, the protein becomes a spiral. It is a right-handed helix, which is alpha helical structure, is your keratin, and because of intermolecular hydrogen bonding, which forms a beta plated structure. An example is the protein fibrion. Okay.